sorry I'm not able to be in Bangkok with you today, but I'm excited to share with you some of the work that we've been doing at TGen. I lead a group at TGen, which is really involved in bringing new technologies to our researchers. And one of the things that we've been really excited to bring into our research institute is the PacBio Onso system. And one of the questions I get asked a lot is, why are we interested in native, ultra high accuracy, high reads? And the answer is really for a number of applications that we're using within our research institute that are focused on the detection of rare populations. So at TGen, we have research programs in a wide variety of areas, two of which are oncology and infectious diseases. And what these two programs share is that we want to be able to detect populations that are associated with a disease in a high background of non-diseased material from very small clinical samples. And this holds true for samples in oncology, both tissue and also in the liquid biopsy space. And it also holds true from a technology perspective in infectious diseases, where you're trying to detect bacteria within a high background of lung or blood or saliva. And we fundamentally believe in our research programs that early detection improves patient outcomes. And in order to detect these diseases earlier, we need more sensitive tests. And that's where we're really interested in bringing the Onso platform in-house, is to understand how that can help us achieve this institutional goal. Now, some people ask, isn't this already a solved problem? There's all sorts of companies and clinical tests to look for tumor tissue and do tumor profiling in oncology, as well as to detect infectious diseases. But we feel that there's a lot of room for improvement in these clinical tests. And in fact, a very recent study in the area of oncology for gene profiling in tissues in a CLIA lab shows that more than half of the samples that this lab tested don't meet the criteria for the size of the sample, where they have too small of a sample collected in the biopsy, or that the biopsy material has an incredibly low, less than 20% tumor content. And both of these are limitations in our current technologies around clinical testing in oncology. In addition, most gene profiling tests from tissue have clinical reporting thresholds of 1 to 5%. And so if we want to have better tests that detect disease earlier, here are 3 opportunities to create better, more sensitive tests. And as we go to these more sensitive tests for all of these application areas and to enable those lower reporting thresholds, this adds a lot of complexity and also artifacts as the sample preparation and the library preparation becomes more and more complicated, because we end up adding PCR cycles because we don't have unlimited amounts of blood. Now, one solution that's come up that you're all very familiar with in this field are UMIs and the large number of other error correction methods to correct for these artifacts after you do the sequencing. But as we go to smaller and smaller samples, and particularly looking into the liquid biopsy space, what we're finding is that these error correction methods not only add a significant amount of complexity to the library prep, but they also require a huge amount of sequencing, which is discarded. And in this recent biotechniques paper, they've estimated that using multiple error correction methods, that these error correction methods require 60-fold deeper sequencing than would be the case for non-error corrected libraries. So even though sequencing is getting cheaper and cheaper and faster, to use these standard techniques that are available means that you're doing a lot of excess sequencing to correct the errors that you're putting into your samples. And that these are only going to get worse as we go to smaller and smaller samples. So this is really the context in which TGen decided to evaluate, does PacBio sequencing by binding, which is the ONSO technology, can we use this to identify rare populations without error correction methods? And I'm going to talk to you about two use cases. The first is detection of drug-resistant TB, and we actually did this in collaboration with PacBio on a pre-commercial prototype instrument before ONSO was launched. And then I'm going to tell you about some of our newer data on the commercially available ONSO system that we've installed in-house. So I want to start by talking about Mycobacterium tuberculosis. So tuberculosis is still one of the major causes of death from infectious disease. Roughly one person globally dies from tuberculosis every few minutes. And the problem is becoming greater and greater as multi-drug resistance or extensively drug-resistant tuberculosis starts to take over and become a larger public health issue. So antibiotic resistance happens when you have bacteria within the lung or the sputum that develop spontaneous mutations that confer antibiotic resistance. And as those antibiotics, as you keep taking them, they're going to kill away all the bacteria that don't have this antibiotic resistance. And that allows the antibiotic resistant bacteria to grow and to take over and become the dominant strain. So you can see that detecting these antibiotic resistant strains very, very early in a background of not just lungs and sputum tissue, but also in a background of non-drug resistant TB is really critical towards treating this disease. It also makes an outstanding scientific pilot project, particularly at a time when the ONSO was literally breadboard number 5 and not at the scale that it is today. And so we wanted to start by looking at two genes within the tuberculosis genome, GIRA-A and CAT-G, both of which have a large number of mutations that are associated with extreme drug resistance. TB is a 4.4 megabase genome. It's got 4,000 genes. It's a challenging genome in that it has a very high GC content, on average at 65%, and it has very well-defined known hotspots for antibiotic resistance. So the experimental design is that we started with tuberculosis plasmids that were either mutant or wild type, and we amplified out the gene of interest that has a known mutation shown here by the red bar. And we converted that into a sequenceable library. We did 3 experiments using mixtures of these amplicons. So the first is that we sequenced these on the PacBio SBB sequencer. It didn't have a name yet because it was pre-commercial. And we did single reads where the mutation was towards the middle of the amplicon, and I'll be presenting that data. We also took those same amplicons and sequenced them on an Illumina MiSeq instrument. And we did the data analysis in 2 different ways. So we did the same analysis as what we did for SBB. So you have an apples to apples comparison where we looked at the single reads and looked for the point mutation. But we also did an error correction method where we require the mutations to be present in both forward and reverse reads, which is a pretty common error reduction method to drive down the sequencing error. So we'll be comparing these 3 data analysis methods. So the first experiment was really to understand, as we mix these 2 amplicons together, wild type and mutant, and we did a titration curve, how low could we go in terms of detecting the point mutation? And you can see that we did extremely well. I think we included some of these low data points as sort of a joke. initially thinking there's no way we'll be able to detect 0.001% of mutants, but let's try it anyway, just for fun. And you can see that for one of the genes, CAT-G, we do detect the point mutation at down to 0.001%. In the other gene, GRA, you can see that we aren't able to go that low, and that we're able to detect down to 0.01% mutant. And I do want to indicate that this was we didn't expect to be able to get down to these really ultra low percentages. And so in reality, we didn't sequence deep enough to have real true statistical significance around these low percentages. But we also wouldn't run an experiment sequencing to 100,000 read depth anyway, due to operational requirements. So to do a deeper dive, actually, I want to go back and talk about one hypothesis at the time for why these two genes behaved slightly differently was that they have incredibly different GC contents. And you can see that in the CAT-G gene, where that mutation, that red bar, is at roughly 60% GC content. Whereas in that GRA, where we had slightly weaker performance, you can see we're looking for something that's in a 70% GC region. And so you can imagine that any sequencing technology is really going to struggle. This was actually a very challenging problem. I do want to highlight that this is several versions of chemistry in the past, and that what we're seeing on the newer chemistry is much better high GC performance compared to what we had on this original breadboard. So when we take that same data set and we downsample it to 20,000 read depth, which is what we do standard in our laboratory, we are able to detect minor variance down to 0.01%. And here you can see that on the left, we have what the error-corrected SBS plot looks like, where you can see that there's high uncertainty in the data, versus what the SBB data looks like, where we're able to detect a full order of magnitude lower than we could on the SBS, and with a much better confidence interval. This is really kind of the slide that really brought home the value of this technology to our faculty when I was presenting the initial study at our faculty meeting. So here I have an IGV plot that compares the data on the MiSeq on the top and the SBB data in the bottom pane. The red sort of confetti-looking dots are all the bases which are a mismatch to the reference genome. So intuitively, immediately you can see that the SBB data is much cleaner than the SBS data. Now you can see at the time, the SBB reads were still pretty short, and this has changed in their commercial onso, and now they're a full 150 base pairs. But the thing that I really want to emphasize is not only can you really look for that needle in a haystack and really find that point mutation down to 0.01% or lower, but it also really reduces the false positives that you're going to have to go and investigate and track down. If we want to look at this quantitatively in this table, here on the right, you can see the dilutions, the percentages of that mutational strain, that needle in the haystack that we're looking for, and look at how we analyze the data in those three methods I talked about. So the top set is the control SBS, so that's on a MiSeq, just the single reads, no error correction. And you can see we detected 1%, and even at 1%, we're getting 30 false positives in the data. When we error correct that MiSeq data in the second set of rows, you can see that gives us a full order of magnitude improvement in sensitivity, and we can detect that point mutation at 0.1%. It really cleans up the data, but we still have false positives that we have to go and track down. But by using the PacBio SBB with no error correction, we're able to detect down to 0.01%. There are still false positives. However, they're significantly lower, and they're equivalent to what you would detect using error correction. This is really, I think, an outstanding result, particularly for a pre-commercial instrument. So the data that we were able to see on the onso pre-commercially led us to purchase the commercial onso and bring it in-house. And I want to tell you a little bit about what we're doing. And as the scale of the instrument has grown from a prototype to a commercial instrument, we've been able to move from infectious diseases into human oncology studies. And one of the areas that we think that this instrument will really provide value to our research program is in the area of liquid biopsy and looking for mutations that are associated with cancer in plasma by looking at circulating or cell-free DNA. So the experimental design for our first project in this area is to take 250 clinical plasma specimens, half of which are from people with a known cancer diagnosis, and half of them are from people who are presumably healthy. These go through a standard shallow whole genome sequencing library prep. We really like the Watchmaker kit. And in this case, we did not use the native library prep that PacBio is offering, just due to the timing of when this experiment started. Once we had our shallow whole genome sequencing libraries prepped, we did two things. The first was that we sequenced these on an Illumina NovaSeq 6000. We also converted those using the Onso library conversion kit, which is a few cycles of PCR to change out the adapter sequences, and we sequenced those on the PacBio Onso. One question that always comes to all of our minds is how fast can you bring a new instrument up into your laboratory? And so that was one of the things that we really wanted to track here. And so I want to share with you our burn down chart of how fast we were able to get through this pilot project. So the first thing was we had to convert all of these libraries to be Onso compatible. So the target that we gave to the lab is shown here in blue of how fast we wanted to have these converted. And in red, you can see the conversion speed. And so they clearly were significantly faster than what they needed to be in converting these libraries. As we were analyzing the data, there's a few samples that performed very poorly on the Illumina system that we have not converted. And that's why that line doesn't go to 0, is we're doing a deeper investigation to understand if we're going to include them in the Onso study. In terms of sequencing speed, the green line shows what we expected to accomplish by running 2 flow cells per week on this instrument. And you can see that we were pretty successful in doing that. In fact, we were getting some weeks where we were doing 3 flow cells per week until we hit about April 15th, when we ran out of flow cells because some of it we were going a little bit too fast. And so you can see that we've now sequenced all of the samples, and in fact, this chart is a little bit out of date, and we have completed the sequencing part of this pilot program. In terms of the data quality that we're getting in our lab for multiple flow cells, we're getting really good cluster density, which is mostly in the range that we expect it to see. You can see that in the beginning, we were tweaking the library concentration a bit up and down to try to maximize it. We would overshoot a little bit, tweak it, drop the concentration. You can see the cluster density fall here between flow cell 3 and flow cell 3B. But it's been fairly consistent, particularly for a new technology. The projected yields are all within its specification. And we also have what we would expect to see with the percent undetermined. Now, this is an interesting metric for us, and we've discussed it a number of times with PacBio. And this number is tends to be on the high side for converted libraries. And although it's consistent with what other labs are seeing, but what they're seeing internally is that with the native library prep, this number is much lower. And it seems to have to do with how the samples are being demultiplexed. And I think this makes intuitive sense to most of us who've brought other sequencing technologies online, that there's just some historical indexes that perhaps aren't playing well together. But I think this is an area where we're going to learn a lot in the next few weeks to help bring this number down. The top bar here is the total number of reads, which tracks very well with cluster density. The bottom line here is a quality metric that basically says that 90% of the bases are at this Q value or higher. So you can see on average over 90% of the bases are having greater than Q40. And you can see that where it gets quite low, meaning only 35, that's flow cell 3, which is the one that we over-clustered. And so there's a clear correlation there. The sequencing quality by cycle is remarkably good. So I've plotted here on the bottom the NovaSeq Q scores for a set of samples, as well as what we're seeing on the ONSO SBB. And you can see that for these 20 samples, on average, we're seeing quality scores of over 55 at the beginning, which is really quite outstanding. In terms of demultiplexing, you can see that we are generating very consistent number of reads per sample. We are generating slightly less than what we generated on the NovaSeq per sample. Some of that is experimental design, as I'm sure all of you know. Sometimes when you give it to the core facility, you get variable amounts of data. We have a very high percent passing filter for the ONSO data. And I think one of the most interesting things to us that we don't fully understand yet, but are very curious about, is that on the ONSO, we're seeing a much lower percent duplication rate than on the NovaSeq. And I think that's, when we've talked with our support staff at PacBio, they're seeing that at other sites as well. So we haven't completed the pilot project yet. We're deep in analysis, but I think that you can see that we've generated a very large amount of very high quality data, and we're hoping to be able to present the results, the biological results of that study this fall. So I want to thank everybody at PacBio and TGen who's contributed to these projects, and I want to thank you once again for your attention, as well as the invitation to come and share our experience with you today. So thank you very much.